going to show you today is our past research that was funded by the Glanzmann Research Foundation and our current goals to get this into the clinic, okay? So first I have to say that the things I say today are my own thoughts, and the NIH supported a lot of the work too. And what my views are not necessarily their views or the views of the medical college. And so that's what we always have to say. I'm president and founder of a platelet targeted therapeutics. I didn't know that you should name a company with just one name, so we call it PTT. The reason I started this company was because when in 2012, when I thought, okay, in order for this to go to people, it has to be protected. Not because I thought it'd become rich off of Glanzmann, right? It has to be protected with patents. And in order for, I thought, for companies to pick it up, they have to know that they have some security that not somebody else is going to try it. So a pharmaceutical company would be interested in. So I got out all my credit cards and charged. I think I told my wife or not, I don't know. But we did ourselves. So these are all the countries, if you're watching on camera from your countries, that we have patents in. South Africa and Europe, Canada, Brazil, Hong Kong, Switzerland, Denmark, India, and Japan. And I just want to say, this reminds me that even though we're in the USA right now, there are a lot of excellent physicians for Glanzmann's all over the world. So if you're in one of these other countries, or if you called me on the phone, or if you emailed me and you said you had a problem, we would look to see where you live and who is the local expert in your area. And we usually go into the publication. So don't think that America is, Glanzmann was discovered in Switzerland. So first described. So let's move on. It was great to be.
The gray one.
Which one?
This one. This one is.
Oh, got it. Okay. Thank you. I know you're up for freeze.
It's
If I go on and on, just look at the title and I'll stop sooner or later. So, okay, so first one, an introduction, talk about Glanzmann. I had many great speakers before me, so I can breeze through a lot of that. I'm just going to pick up some stuff that I thought you might also know. The history and diagnosis. We'll go through that quickly. The history of genetic therapy, I'll talk a bit about it. And gene therapy overview. And I'll start with platelets. Dr. Valentino gave a nice, but these are the old diagrams that we used to use showing the many receptors on a resting platelet is round. And then in your body, you have epinephrine and they become adhesive. When you get an injury, lots of things are going on. The platelets become activated, secrete agonists and serotonin. And alpha-2b beta-3 is up here. It's involved in binding to fibrinogen, and it helps platelets to clump together and seal off the vascular injury. And that's what's missing in 2B3A in GT. So I have to remind you, we're talking about Glanzmann today, but about 1 in every 20,000 people has a rare inherited platelet defect. And usually when you have a platelet defect or a protein on platelets, you bleed. So you may have heard of some of these. And if you know the protein that's affected, some of these are more common than others. Scott syndrome, Wiskott-Aldrich syndrome affects other cells, but so these are called ultra-rare disorders. But if you add them all up together, and then over here, you don't have to remember all this, but it tells you what part of platelet function is disrupted. And Bernard-Soulier syndrome is popular, is well known. And usually Quebec platelet syndrome. And so Glanzmann is right here. About 1 in a million, we estimate. I may be underestimated. The protein names originally were glycoprotein, GP2B3A. Then it was alpha-2B beta-3. And now it's ITG, A2B, ITGB3. So those are gene names. So, every generation likes to updated. So I'm kind of from the older generation, or was trained. So I may use these names interchangeably, but it's either 2B or 3A. You heard about, but you haven't seen him. This is Eduardo Glanzmann from Switzerland. And his paper came out in 1918, if you think about it, and it was in a German pediatric journal. What was going on in 1918? World War I. So there was a lot going on. And we don't talk about that too much, but there was a lot going on at that time. So for him to get this published, then I would think had to be a big deal. So we've already talked. He's from Bern, Switzerland. And his first patient, the platelets couldn't clump in a test tube. And I'll show you a blood clot later. They couldn't retract the clot. And that's how he noticed the person. And he called it thrombasthenia, was said by Michelle, means weak platelets. Normal platelet number. And so many people showed cartoons, alpha 2B, beta 3. And the one point is they're on separate genes. So you can either have an alpha 2B defect or beta 3 defect, and they form a complex. And if either one is defective, the whole thing doesn't show up to the surface. So either one messes up the whole works. And pardon me, I'm from Wisconsin, so I have a jargon from Wisconsin. But anyway, so Alan Nurden published many great platelets or papers on Glanzmann Thrombasthenia. This is just one of the nice, where he kind of put up many mutations. You see the mutations are all over the place, one gene and on another. So when you're thinking about gene therapy, you think of some gene therapies, okay, I'm going to fix, if I did gene editing, I might just fix that one or this one, and each one is a new drug. But then our idea is to replace the entire gene with a normal gene, but instead of trying to fix each mutation. And we do have a website at the Medical College of Wisconsin that Dr. Kohler, fermenter, asked me to take over. He started it at Mount Sinai. And if you go there, you can find the mutations. You may even find yours if some people in the room may know that there's been reported already, and it's all published. And I say generous contributions from patient, family, and friends, because I do use some of the money from GT to help, because I figured that's what you want. And any money I get from GT, I use for GT, okay? Every year I have to pay some money for a certificate or something. It's not a lot, but I thank you. Okay, my first patient. We used to name the patients by their initials. You can't do that anymore. It's against the law. It's the HIPAA, right? Protect people. But this is LM. I got a little sample of blood in 1991, which was a long time ago, more than 30 years, of LM. He was a little French boy. And his family, the blood was flown all the way from France. And I found out that his mother, okay, this is for GP2B and 3A, and his parents, and this is normal control, he had no 2B and a faint amount of 3A. So that gives you a hint right there that the problem is alpha 2B is missing. So I figured that out, and I got my PhD, and I got Peter Newman. There I am, 1994. I had more hair. I went to my grandma's house, and my aunt was doing dishes, and she dropped the dish, and she said she died and thought to went to heaven, and Jesus walked in her house. So I got rid of that haircut. Okay, I didn't want to scare my aunt anymore. But anyway, so I was happy with characterizing the defects, but then I thought, how is this? They always come out and they don't work. So I want to try to fix them instead of finding something wrong, because there's always something wrong, right? So I said, I want to do gene therapy, and that's just when gene therapy was becoming very exciting. So I think this is I went to Georgia, as was said before, from 2001, 2008, to explain what we were doing with the donations that we were getting from the fundraisers. This was at a Catholic church that was decommissioned, and it was turned into a casino hall. And I think you can recognize some people here. Emily and Julia. There's other people in the room here. Peter, where are you? Is that you? Okay, we could sit here all day pointing people out. This is me, and this is my wife, Savvy, who's in the room announcing people. And so this was, and I remember I was teaching Julia how to play blackjack. And Helen said, "You're corrupting my daughter." I said, "It's just for fun." So anyways, then Julia got to meet President Bush, which I never did, but I'm sure it was very exciting, and I think it really helped push the field forward. Okay, so we already talked about diagnosis, so I'm going to go through this really fast. A lot of kids at age 5, a lot of times people are misdiagnosed. They look at medical history, presentation, laboratory, prothrombin times. Okay, so you've seen all this, and so I'm not going to go through it anymore. Michelle gave a very good talk about it yesterday. If you have, then Lorena's going to talk about mutation analysis next. So the standard of care, you heard already. They use super glue sometimes, fibrin sealants, TXA, you heard about all this yesterday. Iron replacement if necessary. I thought I had on there. Oh, I got it on the next screen. Dr. Salted pork girls, okay? That's because I went to the meeting in Austin and I learned about it there. So every time I come to your meetings, and it's our meetings, I learn more and more. So I'm very grateful. I learned a lot in the last 2 days, a lot of great speakers. So, as a treatment, bone marrow transplant was talked about. There's risk of complication, but it can correct GT, and it has corrected GT in some people. And I found one of Alan's papers. Thank you, Alan. He tracked people from 1985 to 2015, and this one I'm sure it's been updated, but there were 43 people transplanted to that time. 35 were still alive, 3 passed away, and they lost track of 5 other people. So it can work, and it has worked very well for some people, so I don't want to knock it. But you are getting a transplant, and the National Registry is in Milwaukee, Wisconsin, the National Bone Marrow Registry, where they match you. I would say that anyone who can't use that, for our gene therapy strategy, I'm going to show you, we use the patient's own cells that we collect from them and genetically modify them and put them back in. So we feel that there's graft versus host, and where the graft attacks the person, and when the person attacks the graft, they can lose the graft. So we hope that we could avoid some of these complications. Okay. So I think I just said that. You get these different problems. You could get infertility. They have been giving, you can either get a mini transplant where you just correct a certain percentage of the platelets, or you get a full blast where the entire marrow that you're getting has to engraft, and you have whole new bone marrow and new platelets. So this is our strategy for stem cells. They're an ideal target for gene therapy. It's been done for many years. It's not our invention. You can safely mobilize people with cytokines, growth factors that will make the bone marrow cells go into their peripheral blood. And then you can do something called a pheresis, which I don't know if I'm going to show you later or not, and then you collect the cells safely, and you can return the other cells back to their bodies. And then you can genetically modify them in the tissue culture environment and reinfuse them. And we wanted to just go into the platelet lineage with GP2B3A because it's not in these other lineages. We didn't want to affect red blood cells or white blood cells or anything else. We just wanted to be in that lineage. So we use a megakaryocyte-specific gene promoter. And platelets are normally round discs that float around in your bloodstream, about very, very small. They break up. They're not really cells. That's why they call them platelets. They break up from these huge, mega means big, right, in Greek, megakaryocytes into these little platelets. And when they become activated, they get very sticky and and go to the wound site. I have to see what I wrote. Okay, this is just overview of gene therapy. So what we're doing in gene therapy is you have the cells, the bone marrow stem cells, you have the cell, the nucleus, and the chromosome. You probably heard about DNA for sure. And what we want to do is use a gene transfer vector, and we use the lentiviral vector. And you put your own gene of interest in there. It goes into the cell, it incorporates into the DNA, and then you put the cell back in, and hopefully it would make the protein for the person's entire lifetime. And that's what we're showing here. You just goes back in, and the cells seem to find their way back into the bone on their own. So we don't have to do much after that. So my goal is to develop methods for genetic correction of inherited platelet disorders. And we're utilizing the GP2B specific gene. I hope I'm not poking you in the eye with this.
Nice.
Please. Keep your sunglasses on. Anyway, where was I? So we're going to replace the gene for expression of normal alpha-2b, beta-3. If you're missing alpha-2b, you get alpha-2b. And if you're missing beta-3, you get beta-3. And it'll pair with the other partner that's not affected for Glanzmann Thrombasthenia. I'm just going to call it GT. Okay, this is what our vectors look like. It's a lentiviral vector. We have one for beta-3, which we're working in our lab on. Savi, Dr. Kukuritaki is working on this one. She completed it. It has to be a certain way for the FDA to accept it to use for gene therapy. I'm not going to get into that. And the other one, the same system to make it into megakaryocytes is GP2B or alpha-2B. And they can't both fit in the same vector because they're too big in this situation. And you probably wouldn't want to add more 3A to somebody who has perfectly fine 3A or more 2B to somebody who has 2B. So we have to do the sequencing of you, a patient, in order to find out which protein is affected, to know that we give you the right medicine, right? So, just quickly, as I said before, we give growth factors. We try this, that's on a mic, it's supposed to be mice. We've done this in mice, dogs, and 2 human patients who volunteered to undergo but not transplant. And you would give a cytoreductive, they call it now, but it's a chemotherapy preconditioning regimen, mild, and you see these nice proteins come back to the surface of some of the platelets. You don't need all your platelets to be corrected, we found out. So now my history. In 1999, with Gil White's lab in North Carolina, we first showed that we could use this promoter to target things to proteins. And then in 2000, so you have to take steps. We treated the first 2 patients who volunteered. They're brave because you have to give them cytokines. You don't know what's going to happen when somebody's cells mobilize and they have a bleeding disorder to their bone marrow. I gave them a box of chocolate to thank them for being in the study, but we had to watch them very closely. I think they got maybe mild headaches you do get some pressure on your bones, but they did very well, which gave us encouragement for the protocol. And then we did a gene therapy strategy in mice that we showed in 2005 that the beta-3 knockout mice were created here at MIT in Boston by Richard Hines, Dr. Tuttleham, and Dr. Kohler, Hermanter. They all formed a collaboration. So the Glanzmann mouse was created here in Boston. And then once they created it, I fixed them. So out of 21 mice, they all did great. And we treated 3 dogs in 2011. It was Dr. Kohler communicated this, edited for us in 2011 we say improves hemostatic function in dogs. And I'm going to show you a little bit of that data. These are the people cells. And one person was from Johns Hopkins, from Paul Bray's group, and the other one was from North Carolina, Gil White's group. If you look at glycoprotein 1B in this patient, it's the normal control. There's no problem with glycoprotein 1B compared to the normal. But for alpha 2B, beta 3, these two people had beta 3 defects, so we put beta 3 in, and you can see these are now glowing after the treatment in these patients. And I was very excited. It was a Sunday afternoon, and I called Gil on the phone. He didn't answer. And I called Peter on the phone because I was so excited. I wanted to tell somebody. Nobody answered the phone. So I just had to keep it to myself. I hung up the phone and went home. Okay. These dogs are Great Pyrenees. They're sheepdogs. And the first dog, they were traveling with a circus, Russian circus in Mississippi. I know they don't have circus anymore, but we used to have circus here all the time. I hope you know what a circus is if you're younger. But I took my son to the circus. You get a picture with the monkey. You get a picture with, all the animals and have hot dogs and everything. And I'm broke by the time I get out of there. And they were selling dogs too. And they sold 2 Great Pyrenees to somebody. And they took the dogs home. And when their teeth came in, they started bleeding all over their rug. So they went to Mary Boudreau at Auburn University, and she recognized it as Glanzmann. And she had to sequence the normal dog 2B3A genes first, and then she sequenced their mother's 2B3A, and she found this insertion of 14 nucleotides on intron-exon borders. So anyways, it was up here. And these are the 4 first dogs. We named this one Ed in honor of Dr. Glanzmann. I won't say he looks like him, but he was the only male we had. So, okay, this is what it looks like on a 2-dimensional if you run the proteins on the blood. And what you really just need to notice is that these are labeled 2B3A, and these are the old system, and they're missing. So that was the early blots of the dogs. And to confirm the protein, because you can have a DNA change, but it doesn't always mean that you're going to have a missing protein or a defective protein. It could just be called a polymorphism. This is what I was talking before about the blood clotting. So if you notice here, there was a litter of 10 dogs, but 4 of them, their blood doesn't retract into a fibrin clot. And that's the definition of the very first definition of Glanzmann Thrombasthenia. So their blood won't clot in the tube. So this is we made this first into a clinically relevant protocol. Some dogs came up to Milwaukee. We did something that we would want to do in people. We mobilized them. We gave them canine growth factors, collected the cells, stimulated them, transduced them with this vector. It's a little bit different than the one you saw before because it has we call drug selection gene, but we wouldn't want to put that in people. But anyways, and the techniques today that since 2008 are much better for percent cells that you can correct and the number of cells you can give. It's just totally improved. And so we gave them the reduced intensity and immunosuppression for a while, and then we looked for gene expression. And these are they're big, hairy dogs, right? And I have to shave their legs before we can collect blood. So what you see here is they have huge petechiae and bruises. And Quinn was showing me a bruise from his diving, and it looked like that, and it didn't. So after we did the treatment for 6 months, we see, wow, this is like a leg commercial for pantyhose or something, right? Their legs are clean. And I had to get permission to take pictures of their legs, but I was amazed because these are like little microbleeds and they're gone. And so we knew something was working even before we drew the blood. Okay, so this is a little bit technical, but I'll just show you. So we did this thing called, I remember Dr. Kano was talking about bleeding times. We did a buccal mucosal bleed time where you just make a little cut on their lip and you twist a paper around, like a tissue, around on their mouth for every 15 seconds until they stop bleeding. And so if you have Glanzmann's, your bleeding time, we stopped them bleeding at 20 minutes with the super glue. And they lost a lot of blood, about 2.5 to 1 gram. the control dogs. And our dogs did the dog A before he was treated. And then after he was treated, you see all the tiny little bit of blood lost, and his bleeding times dropped from 9 to 10 minutes. And dog B, which we followed for 4 years, and you know those little puppies, I didn't treat the puppies because by the time I knew what I was doing, they were already maybe 4 years old. I didn't want to treat them until I knew that I could handle their cells in the tissue culture, because this is something when you go from a mouse, and all the mice were happy we could cure them, but when you go from a mouse to a dog, you got to be really sure what you're doing is right, is going to work. I'm pretty sure. So you can see the bleeding times dropped to 7 or 8 minutes in this dog, 4 years out. And the dog C was the best, and he was down to 6 to 4 minutes, which is near normal bleeding times. So they stopped bleeding on their own. And he's 5 years out. So I'm not permitted to compare dog years to people years. But 5 years in a dog, I think they went out to 6 or 8. And we let them go live on a farm in North Carolina to live the rest of their lives out after our study was over, because you want to see how far they can go. They all did very well. And if you can get a bleeding time around this in a human, I would think, I think we can do better than that, or at least as well as that, with the new technology. I'm hoping for that. And Taylor asked me to show you. One dog, as you can imagine, never saw, we put human alpha-2b in there, which has about a 95% homology with dog 2b. We didn't have dog 2b, so we put human in. And so it paired with the dog 3a, and he developed this huge immune response to the protein. I'm not crying because I'm just delighted. So what do? We did what Nathan was talking about the other day. We gave IVIG, and it knocked it right down. And we only did it for 2 days, 2 treatments. And it just knocked it right down, and the body accepted it. And he went on forever, or for the rest of his life, just fine. Or she did, I should say. So, hematopoietic stem cell gene therapy, encoding the replacement gene, worked well in the human patient cells, ex vivo. It worked very well, safely in the marine model of GT. It also safely restored 2B3A to the surface of canine platelets and showed improved platelet function and established hemostasis, thus, well, we say correction, of the bleeding in mice and dogs, or restored hemostasis. This indicates the feasibility for targeting platelets, human platelets, with genetic therapies for paving a better way for management of humans with inherited bleeding disorders, Glanzmann and all the other ones you saw that we know, also deserving of a treatment. So let's talk about the trial, and then I'll finish up. I asked Missy to take a poll. I'm not recruiting anybody. I'm just finding who's interested. I'm not permitted to recruit. But we just were kind of curious to know on the GT website how many people, what their severity of bleeding was, and how many people would consider gene therapy if it was available. So here in the very severe group, it was 1 in the United States. And I have to tell you, if we're using NIH dollars, we have to, we tend to, unless it's written in the grant, we treat in the United States first. And not to say that we wouldn't make this available to the entire world, okay? And so there were 22 people and 13. So we have a number of people from the United States that are interested in this, which is very good when you make a proposal. And I'm guessing that we probably would stay away from, you want to treat somebody in the severe category when you're doing a gene therapy protocol. Then I did put female there because, and you have to, I don't think you heard this, but you may know it, we talk about hemophilia A and Glanzmann. Hemophilia A occurs on the X chromosome. That's why it's mostly in males, because males only have one X chromosome and one Y chromosome. While females, there are females with hemophilia A, but they have to inherit the X chromosome, factor VIII damage on X chromosome from the mother and the father. But for Glanzmann, it's on chromosome number 17. And therefore, females can get this, and males. And there's 3 different ways that people are talking about correcting or improving in gene therapy. One is called induced pluripotent stem cells, where you take the cells out and you, Morty Pons' group showed that you could modify the cells and then immortalize them and leave them in the freezer and correct them. And then one way might be to give the people cells, but you don't have to give them chemotherapy, but they would only last a few days or weeks, and then they would be gone. But you could go reach in the freezer every time you needed one. Another way would be to modify the bone marrow. We tried that in the first dog. We took bone marrow because bone marrow is a very rich source of cells. But when we tapped the dog's humerus, she bled for days. And so I said, no, that's not a good way. And so we did the pheresis, and these dogs, a normal person, when they come in pheresis, they just sit in a chair, fall asleep, or watch football or something, or they play video games. But dogs would jump all over the place, so we did have to give them some anesthesia. But anyway, so we transduce the cells with the vector, and we give it pre-transplant conditioning with mild chemotherapy agents to make a space in the bone marrow for these cells that we're putting back in, so they can go and make a new home, and then transplant them. People are in the hospital for about 20 days, and these cells should become mature megakaryocytes, break up into platelets, and be corrected. We do have a hemophilia A trial going on with the same method, but factor VIII instead of 2B3A, and therefore I took our protocol for hemophilia. And you have to know, I got to back up for a second. Do I have time? We have, I called the FDA in 2011, and I said, okay, I want to do gene therapy for alpha-2B, beta-3, and factor VIII. They said, nobody's ever proposed that before. And I was kind of naive, but that's 3 different drugs. And then I said, and they said, are you a medical doctor? I said, no, I'm a PhD. Nobody's ever done it if you're a PhD. Now it's changed. You can be the PI if you're a PhD. You just got to put together a great team. So anyways, there are inclusion and exclusion, and these are taken from the other one. We would pick an adult. If you can treat adults, you should, and you will. You'd have to. If somebody can survive to adulthood, you always have to treat adult first. So we'd have the first volunteers would have to be greater than 18 years old or greater, with severe GT, history of frequent spontaneous bleeding events. You have to be diagnosed with severe GT by looking at your alpha-2B, beta-3 levels and your genetic defect. And your platelet aggregation has to be abnormal. And you have to be on prophylactic therapy after the transplant for a while you're in the hospital, because when you do a bone marrow transplant, it goes up with time, and it can take maybe, what I've seen, 6 months before full effect. So you'll still be on your regular treatments to keep you from bleeding. You may get platelet transfusions, but in that case, you're probably immune compromised, so your body wouldn't reject the platelet transfusions even if you had an inhibitor or antibody. But there are certain things that can be done, but the regular prophylactics have to be available when you're treating somebody, because for a human, we don't know what to expect. So, and then the exclusion criteria would be somebody who can undergo peripheral blood stem cell with the growth factors. Given these growth factors have adverse reactions, you can undergo pheresis. We had one dog where the veins were too small and the machine kept stopping, and that's just unfortunate, but we couldn't treat her. And you can't have any other significant organ dysfunction or, some problems that would prevent you from being in the trial. And I don't have a clinical trial, but this is where we're headed. And so Len was telling me, he had a paper out about do no harm. So we want to maximum benefit with minimal risk and maximize the benefit of improved hemostasis, but reduce the risk of pre-transplant conditioning regimen. I don't want to correct GT and give you another problem, trade one problem for another, okay? We want people to be better off than they were when they came in. And we say it's 3 steps. Bone marrows are collected as an outpatient. You return in 4 weeks. We would treat the cells, freeze them, and then do all quality control to make sure that they're viable, that they're not contaminated, that they have good gene expression, all that before we would give you the conditioning regimen to put them in your body, because it's not worth giving you the conditioning regimen if you're going to be, it's not going to work. And then we monitor you for, in the first year, it's like every 3 months, you'd have to come in and get your blood drawn. And then I think after that, it's every 6 months for the next 5 years. And then, but the FDA requires us to monitor for 15 years. So, I think I already showed this. Oh, no, I didn't. Okay, this is just our technical protocol, and I think we'll probably skip this because this is the actual protocol that I just told you about. And clinical trials have inherent risks. Okay, there's risks for any treatment you're going to get, and this would all be explained to you by the doctors, and the patient advocates and the social workers would go through this with people just to make sure they understand everything. And it does take, our first hemophilia patient is 15 months out now and 12 months without his prophylactic with this kind of protocol. And we're going to show that soon, but I can't talk any more about that today. But just to tell you that we would follow a similar protocol and just make sure everything is well. If anything happened that wasn't normal, we would have to report it. Okay, so I think this is my next to last slide. So we've had 20 years of working on this with Helen and now GT, the new, and we characterized the patients in the USA, genetic, and Lorena's going to talk more about characterizing genetic defects. The FDA required us to update the vectors, and that took some time. We have to determine your specific defect. I'm writing right now the phase 1 trial, and that's what's going on, and there's a lot of paperwork. It's essential for us to secure funding. As you know, the dog work was expensive, and the human work probably we would propose to do 2 people as a pilot study. I'm guessing it would cost about $5 million. But to go from dogs to humans, it's like selling lemonade at my son at the front corner to selling potato chips in a factory where they have to be, everything has to be standardized and checked and quality controlled, and it's a very expensive process. So we are preparing to submit an FDA, it's called a pre-investigational drug, to have a meeting with our protocol that we're designing right now. And the clinical trial phase 1 study would start once given approval at the regular IND, investigational drug. And so this is my team. Barry Eapen is the bone marrow transplant doctor. I call myself the study coordinator. Right now I'm the co-PI of the study. And Lynn Malik is the lead hematologist at Diversity in Wisconsin. She works at Diversity but this is for the hemophilia group. We have our own bone marrow transplant center, own bone marrow processing center that the group came for the site visit, went through. We work with Miriam Armant at Boston Children's, and UPenn does an analysis of the cells to make sure it didn't cause a mutagenesis. You have to analyze this, where the gene inserts into your body. And Gil White and Bob Montgomery, and these are all people that helped us through as consultants now. And I got a lot of help from Harvard, David Williams, and a lot of great people, because I'm in the Midwest, right? So we in the both coasts. And a lot of support went through this from many different places. I say generous philanthropics from the Glanzmann Research Foundation, also Jamie Swain and other of you who sent me checks in the mail. I was happy and surprised to see them in my mailbox at work, but all that gets put in the Glanzmann Fund. So I try to send out thank you notes each time if you send them directly to me. And this is for the hemophilia clinical trial going on right now. You can see a lot of NIH support. We would hope that, this is my lab, the Sabi, working with Glanzmann 3A. Lily works with hemophilia. Juan works Glanzmann 2B. And Shannon did, this is a Cobe Spectra apheresis unit, an old one, the newer models, but they're basically like looks like a wash machine that spins around and separates your blood, and then a little straw sucks it out and puts everything else back in your body. And this is the group that you may know: Missy, Peter, me, and Taylor in my lab. Oh, and that's it.
So once corrected, you are now considered a carrier of GT for the rest of your life?
Wow. So like I said, we don't like the word corrected. Once you have restored hemostasis for a long, long time, I guess you would be a carrier, because we're not going to be correcting your germ cells. So some people did think that their children, they could then have children without the GT, but it's illegal for us to tamper with your eggs or sperm to genetically modify them. That's illegal at this time. We do tests to make sure that that doesn't happen. So I guess in a sense, you probably would be a carrier if you were to have you have GT, but I mean, genetically you have GT and you can pass it on, and both of your genes are affected. So technically I would say yes, you carry the genes that could cause it.
I think that was my only question.
Okay.
So stay here. Is there any different in terms of passion program design patients now? Patients.
I don't know. I know that there's several gene therapy trials going on. We didn't invent gene therapy for sickle cell anemia, which is female and male, beta thalassemia. Let's see, that use lentiviral vectors, severe combined immune deficiency, both male and female, done in children. A lot of times they do children if they won't survive, and therefore you can do male and female. I think female is a little bit a different strategy because you have to, like I think Nathan said, you have to collect the, let's say eggs, over whatever. I'm sorry, and from women before you do the experiment.
Yeah, I just think now there's possibly.
But for hemophilia A, definitely mostly men, but there are females. And from what I know, most of the females are pretty mild, aren't they?
There's 75 severe women.
75 women with severe hemophilia in the United States currently.
Well, maybe we should open it for women.
Yeah.
So, there's no reason why you
We
should.
should open it to women.
Yeah. Wow.
There's 2 with inhibitors, by the way.
You know what?
Okay, good.
I'm going to bring it up at the next student. But that's why I put a woman up on the Glanzmann was. But thank you, Len. I'm glad you came.
No.
I have one more.
Uh-oh.
What if you have a mutation on the 2B and onto the 3A?
You know, I'm probably sure that somebody does. I've never heard of somebody that got 2B and 3A knocked out, because you would have to get inherited 2B, 3A from, well, I don't even know. These, that we'd have to think about that.
You can be a composite heterozygote, right? Because you can have a mutation coming from your dad and a completely different one from your mother. So that it is likely that this could happen, yes. But then you will need
But then we
both.
would have GT from a 2B and a 3A.
It depends how severe the variant is, right?
So we'd have to take that on a case-by-case basis.
Now, I do know that there are some out there who do have 2 mutations at the same time. Would the approach be similar, or would you have to change it up a little bit?
Well, the two genes are both on chromosome 17 right next to each other.
Mm-hmm.
So if, let's say, you got some kind of hit with a meteor and your DNA got damaged, it could potentially happen. It's not common, but maybe Lorena would have to help me on that one. I would say they probably wouldn't be our first choice for our gene therapy because, like I said, alpha-2B and beta-3 are 2 different drugs. They're considered by the FDA to be 2 separate drugs. And so we would have to give them both, and that we couldn't do that at the time. Right now, we just want to prove the proof of concept that you could improve hemostasis in a person with Glanzmann's using this protocol. Safety is always number 1, and efficiency. And then phase 2, move on.
Right. Then you will have to treat it separately because, yes, they are, even though they're together, they are controlled by different genetic promoters, etc. So they are treated separately. Okay, so I am Lorena Huitrago. I am a PhD, so I'm a fake doctor. So nothing what I'm going to say here is supposed to be taken as medical or clinical guidance. But I'm really, really honored and very happy to be here with you because as a scientist, we normally get some samples from you guys, also blood, and we research, but this is for me the first time that I face patients, and it has been very humbling and has been very inspiring. And I hope I go back to the lab to work to get better treatments and be creative and try to do something better, right? To help with Glanzmann Thrombasthenia. So I come from New York, from the Rockefeller University, and I'm going to go through my talk, going very slowly and back to basics so that we're all on the same page and we understand the concepts that we're going to be talking about. So I'm going to go back to like high school biology for all of you guys, just to remind you that so we have all our cells, and actually maybe you do know this, but there's only 2 cells in your body that has no DNA, and one of which is platelets. Platelets has no DNA. Red blood cells also have no DNA. All of the other cells in your body have DNA, and they undergo a process of transcription and translation to make protein. So we start with DNA, and we have the typical drawings of the DNA. This goes into something called the RNA polymerase, which reads this and translates this into something called messenger RNA. And this message is now go into the ribosome, which is a little very powerful machine in your cells that reads this RNA, and in a process called translation, it makes a protein. So we go from DNA, mRNA, and this creates amino acids, and the union of all of these amino acids makes a functional protein. So our DNA is mostly composed of nucleic acids, and we have 4 types of nucleic acids only. And the combination of this nucleic acid is what we call the genetic code. So when you go and read this, it's like literally reading a book made of 4 letters, a very boring book. but creates a huge amount of diversity. So hidden within these genetic codes lies what we call the triplet code. And it is a series of nucleotides that determine a single amino acid. So your DNA is read in groups of 3. This is what we call codon. And depending on how they are combined, these 4 letters can be combined in different ways. They will now make a different amino acid, okay? And some of those also tell the DNA to stop. So we have stop signals, or we have amino acid signals coming from this DNA, or these codons, as we call them. So if I can do this. So every time a cell divides, it has to make a copy of this DNA. And this process is called replication. And DNA can be altered by a number of factors, including environmental influences such as exposure to UV radiation or certain chemicals, and changes that occur when a new strand of DNA at the time of cell division is called a spontaneous mutation. And a change in one single amino acid is called a point mutation. But changes in the DNA happen in many different flavors. And you can have a point mutation, or you can have pieces of chromosomes. DNA is organized in chromosomes, so you can have pieces of chromosomes that are altered by deletion, duplication, inversion, or rearrangements. And so this usually affects many nucleotides at a time. No, that is one. So the human genome consists of 3 billion nucleotides. So I was reading earlier that it will require somebody to be writing 60 words per minute, 8 hours a day for 50 years to type all the DNA that we have, all the nucleotides, right? So changes in just one of those 3 billion, right, can result in dramatic physiological variation. But this is also how evolution works, and this is also how we create variants that are selective for positive qualities in our species. So a single nucleotide change can alter only one amino acid in a protein, but the result of this variation can dramatically alter the function or the expression or the phenotype of this protein. So what is a genetic variant? A genetic variant is an alteration in the most common DNA nucleotide sequence. And the term variant can be used to describe an alteration that may not affect the protein, so it's benign. We may cause a change known to produce a disease, which is pathogenic, or it can produce a change that no one knows exactly what it will do, so it is of unknown significance. So now we're here to talk about integrin alpha-2 beta-3, which you all know. And as Dr. Wilcox said, the nomenclature of this has changed, but nowadays we refer to it as integrin alpha-2 and beta-3. Alpha-2 is a subunit and beta-3 is another one. So the gene name for alpha-2b is, as Dr. Wilcox mentioned, is ITGB2A, and the gene name for beta-3 is ITGB2-3. So I will be referring to those names when I'm talking about the gene, but alpha-2b or beta-3 when we're talking about the protein. Alpha-2b and beta-3 are members of a large family that are called the integrin family. And this is made of 24 different receptors that are expressed in many of your cells. Alpha-2b beta-3 is unique to platelets and megakaryocytes. So alpha-2b is only matching with beta-3, but beta-3 can match to other integrin subunits to make other receptors that are expressed in other cells as platelets. But alpha-2b beta-3 is only expressed in platelets and megakaryocytes. And as you know, alpha-2b beta-3 is required for platelets to clump together and stop bleeding, which is what we refer as hemostasis. But platelet clumping also contribute to clogging arteries and causing heart attacks and stroke. So in a sense, we know a lot about alpha-2b beta-3 because of the two kind of sides of the balance, right? On one side, if it doesn't function, you can bleed, and it's essential for your survival. But if you have a lot of this clumping, you can have heart attacks or stroke. So knowing how this protein works is incredibly important for our survival. And alpha-2b beta-3 is so important that there are actually 3 approved medications by the FDA to treat that are going to bind to alpha-2 beta 3, and they're used to treat heart attacks. And usually they're given to people who are undergoing opening of the closed artery, so that they don't clot while they undergo the surgery. So I think you've seen many kinds of this drawing before, but here we go again. So platelets are really, really beautiful, amazing cells. I have been researching them for many years, and I'm in awe of how many amazing things they do. So they are really engineered by nature to quickly respond to injury. And I don't know if you guys know this, but platelets are a mammalian adaptation. So before mammals, there were no platelets. So this seems to be related to the fact that we have placental birth, which of course is a huge challenge, and we really needed a system to keep us alive. So platelets have an array of receptors for different agonists that are in blood, and these agonists are usually produced when you have vascular injury. And here you have our main character of today, alpha-2b and beta-3. So each platelet has approximately 80,000 copies of the receptor. So maybe this doesn't mean a lot to you, but this is one of the highest expressing proteins in our body. I think none of the receptor is expressed as much as alpha-2b beta-3. And when alpha-2b beta-3 is activated, the platelet is activated by any of these different agonists, then alpha-2b beta-3 is usually closed. And now when signals from these receptors coming in from the inside of the platelet lead to this receptor to open. And once it opens, it can bind fibrinogen, which is abundant in plasma. So fibrinogen is always there. What changes is the receptor conformation, allowing it to bind or not to bind. Once this binding occurs, you get platelet aggregation or clumping. And then through another pathway of the coagulation cascade, you produce a protein called thrombin, which converts fibrinogen into something called fibrin, which is something that makes the clot really strong and sturdy and withstands shear flow, etc. And this is what we call thrombus formation. And alpha-2b beta-3 is involved in both the binding to fibrinogen and to fibrin, because these two proteins are 98% similar. So what can happen to the genes now? We're talking about ITGB2A or ITGB3 to lead to changes and variations in it. So here we have the mRNA sequence without any change. And I'm just putting alpha-2b beta-3 here, but this is just a regular gene. So again, we go to these triplets, right, the codons, and each one of them signals to create a amino acid, in this case, methionine, lysine, phenylalanine, glycine, and then stop. But sometimes we have a base pair deletion. So we have something called frame shift causing extensive missense, meaning that there was one of these nucleotides was what missing. So now instead of having the amino acids, we change it. Instead of a phenylalanine, we have a leucine. So now the protein is different. Sometimes this change in having an extra or a missing amino acid, instead of signaling for a different amino acid, it just changes signals to a stop signal. And this is what we call a nonsense variation or mutation, because the protein is no longer produced. And sometimes we have 3 nucleotide insertions, so this changes the frame shift of how the polymerase and how the ribosome is reading it. So it inserts or deletes full amino acids from the protein. And it's worth noting that not all variants will interfere with expression and or function of the protein. Some variants are well tolerated. So genetic alterations to alpha-2b beta-3, as you know, so I will go through this quickly, can lead to Glanzmann Thrombasthenia, and by decreasing the production of the protein, so the expression of the protein is altered, and this is a qualitative defect, or sometimes you have a change that leads to functional defects. So here you see that sometimes you have this integrin that looks a certain way. When there is a second change, now this one looks a little bit different. Sometimes this change is well tolerated and nothing happens. Sometimes this change changes how the cell can make this protein. So usually because proteins are kind of tridimensional structures, So if it is not folded properly to make the right structure, it cannot be produced, or it cannot be matched to alpha-2b or beta-3, so it cannot go to the surface of the platelet. Sometimes the change is different, so this does not affect this step. The protein can be made and can be produced, but it has a defect in binding its ligand fibrinogen. So it cannot bind, even though it's expressed. So depending on how this receptor, this change affects the receptor of the protein, you can have type 1 Glanzmann Thrombasthenia, where you have less than 5% expression. You have type 2, which you have 5 to 20% expression of the receptor. Or you can have type 3, which is the qualitative kind, in which the receptor is expressed, usually in decreased amounts as well, but the receptor does not function correctly. So these are the different changes that can happen to the protein that end up in having Glanzmann. So Glanzmann Thrombasthenia, and due to the explosive growth of the human population, so in the past 2,500 years, we went from being 50 million humans on Earth to be 7 billion, right? So this predicts that a large number of variants enter the population relatively recent. The frequency of GT in the general population, as Dr. Wilco mentioned, has not been really established, but it is rare, except in groups in which the variant entered the population some time ago, and mating within the group was common. The first person to have a specific variant that produces Glanzmann is called the founder, and some studies have indicated, have followed these variations in the DNA and have found out that these variations entered the genetic pool 300 and 600 years ago. So this is recent, and that's why it's also rare. I want to quickly mention that changes in the gene of ITG2B and ITG3A also leads to 2 other different disorders. So, because I will be talking about this later. So changes in alpha-2b beta-3 can also lead to a disorder called alloimmune thrombocytopenia. In this case, usually the sequence change is benign. The protein is produced normally. But what happens is that the mother protein looks different than the baby's protein. So now the immune system of the mother usually recognizes this protein as foreign and creates an immune response to it. So it is now tagged by an antibody, and the platelets are now recognized by the immune system. They are destroyed. So these babies are usually born with low levels of platelets, and that's the name thrombocytopenia, which means low platelet count. And another one, and usually this change happens, and you will see this a little bit later, happens in the area of the protein that is linked to the inside of the platelet or of the megakaryocyte. So changes in these specific parts of the platelet doesn't allow the integrin to kind of hook to the cell, and then this leads to the disorder called macro or anisothrombocytopenia. And macro is because usually these platelets are pretty big, but these defects are now at the level of the megakaryocyte and platelet production. So the same genes, the same kind of changes leads to 3 different conditions, 1 being Glanzmann and 2 natal alloimmune thrombocytopenia or macro anisothrombocytopenia. So alpha-2b beta-3 has been studied extensively by many, many labs in the world. And we do know a lot about this integrin, which is something that is very positive because it allows us to understand how it functions, how it works, and to actually think of things like what Dr. Wilco is doing of gene therapies. We know a lot about this protein. We know how it looks, we know the atomic level, and we know all the different domains that it has. And I just have this drawing here because later on I will just talk to this about this. But we know that it has a cytoplasmic domain. This is where it links to the inside of the platelet, transmembrane, and this is the part that is outside the platelet and that binds fibrinogen and allows the clamping of the platelets. So now I'm going to switch gears here and talk a little bit about next generation sequencing. So next generation sequences is a technique, a technology for determining the sequence of DNA of or RNA, and to study the genetic variation associated with disease or other biological phenomena. So using next-generation sequencing, an entire human genome can be sequenced in just one day. But if we think in contrast on how sequencing was done before, that was called Sanger sequencing technology. So for example, to decipher the human genome, they require 10 years. And now we can do this in just a single day. So DNA sequencing platforms that are now widely available reduce the cost by several numbers of magnitudes. So before it was incredibly expensive to do sequencing of one gene, or imagine just the whole human genome. But now this is affordable and it's commonly done. So today, next generation sequencing is a ultra high throughput, accessible and affordable and a rapid technology. So this is just a cartoon showing you how next generation sequencing works. So you basically get DNA from any individual or samples, and then you cut this DNA in many different pieces, and you create DNA fragments that now go and bind to something called an adapter, and this create a DNA library. This is then put into one of the very sophisticated sequencing machines that we have nowadays. And in a matter of hours, you have your nucleotide sequence. And this undergoes to heavy processing of data analysis, etc. But at the end of one day, you can have the whole DNA of one individual or sample sequenced. So next generation sequencing is really transforming our understanding of genetic human variation. And it is providing profound insights into the impact of both inherited or new variants on human health. But at the same time, the data that is coming from these studies present serious challenges in providing information to individuals who are found to have variant forms of any specific protein. So in the near future, we will all likely have access to next generation sequencing, so likely at the level of our own DNA or the DNA of our future children. We are easily going to be able to get a sample and undergo the process of sequencing. But the question is how this knowledge will impact clinical practice and personal decisions. So now I go into our research today. So today I just want to share with you the experience that we have that we got by analyzing next generation sequencing data comprising variants that alter the amino acid composition of the platelet integrin receptor alpha-2 b beta-3. And this is coming from 3 databases. So we did not get this DNA from specific donors of people. These are just big databases that are coming from different places in the world. So we used Human Genome Mutation Database, 1000 Genomes Project, UK 10K, Whole Exome Sequencing Project, UK 10K, Whole Genome Sequencing Project, the National Heart, Lung, and Blood Institute Exome Sequencing Project. And all of these data sources combined had 32,000 DNA sequences derived from approximately 16,000 individuals, because people have 2 copies of most alleles. So we have 16,000 individuals giving us 32 DNA alleles to study. So I will go quickly through this. So these databases are just, so each country and several institutions are gathering these efforts into make publicly available all the information that they're getting from individuals of the country or specific diseases. And we use the one coming from the UK. And these both databases are coming from UK initiatives. And this one is coming from the NIH. So in the US, this is also happening. And this database is special from the others. The others have patients and or healthy volunteers. This one is specifically attempts to collate all known gene lesions responsible for human inherited disease. So this one is where most of the GT variants are found because they are disease causing. So what we aimed by doing this analysis is to answer two basic questions, right? So when we look inside these genes and we see, okay, so some people have changes, some people have variations. So what can we learn about Glanzmann Thrombasthenia by analyzing these big data sets? And also, can we provide some guidance to patients and to clinicians on how to use this information, right? I know some of you have, the specific mutation, etc. But and now what, right? What do we do with this information? So I just want to show you how we analyze this data. So I go back to my drawing. So again, we have a lot of knowledge of alpha-2 beta-3, so we decided to categorize the different variants that we found based on the place on the integrin that it was found. And just as a reminder, so proteins have like a long list of amino acids, one to each other, and we can start with amino acid 1, and then we go down to however big the protein is. So alpha-2b and beta-3 are pretty big proteins. They usually have around 1,000 amino acids. So the number that you see on your report sometimes, or the number that you see here, is the position from 1 to 1,000 where we found the amino acid change. So we curated all this data coming from these databases, and we went to see, do we know the effect of the mutation? We went into the literature to see the reports, and based on that search, we identify some of those causing Glanzmann Thrombasthenia. We did know in some cases, based on the reports, whether it was a homozygote, meaning that the two copies of the gene had the same change. A compound heterozygote is what we were discussing before, that you have two mutations, but different mutations, and one can be in alpha-2b, one can be in beta-3, or both on alpha-2b and beta-3. And based on that, we also say, based on of all the research we did, we were able to categorize the defect. So we would say, is this a surface expression defect? Is it a ligand binding defect? Or is it a functional defect? And we categorize the Glanzmann variants in presumptive or confirmed. Confirmed are the ones that have undergone studies in a lab. So many of you or have read reports, or even in your case, you give your sample because of the phenotype and because of the clinical diagnosis. They say it's Glanzmann Thrombasthenia. But sometimes those can be wrong. So unless they were proven in a lab in which we engineered the mutation and we made sure that it was causing the defect that it was said to be causing, we're calling that presumptive. Because this is a big database, so we don't want to include things that might be, of course, misdiagnosed. So unless it was confirmed in the laboratory in a recombinant system, meaning that somebody in the world engineered that variant into the gene and expressed that in a cell and proved that it was Glanzmann, it was called presumptive. So what did we find? So we found 319 missense variants identified. And from the confirmed Glanzmann Thrombasthenia, we have found 52, presumptive 59. We had 20 that were causing neonatal alloimmune thrombocytopenia, and 5 that were causing macro or anisothrombocytopenia, and 114 that were novel. We don't know what these variants are doing to the protein. From the ones that are Glanzmann Thrombasthenia, 56 of those were identified in alpha-2b and 55 in beta-3. So from the novel variants, we found a total of 114 variants in alpha-2b beta-3 and 68 in beta-3. In total, 11.2% of the 1,025 amino acids in alpha-2b and 8.7% of the 784 amino acids in beta-3 were variants. So we had changes in that percentage of the protein, and only 3.8% of the variants were present in more than 0.1% of those analyzed. So this indicates how rare these variations are. And this is a model that we created, and this is alpha-2b beta-3. I think you have seen cartoons many, many times, but this is actual how the protein looks. This is coming from the crystal structure. So here is the ectodomain, and this is a big bulky protein that undergoes a lot of conformational changes. This is the transmembrane domain, and right here you have what is inside the platelet. The dots you see that are green are Glanzmann Thrombasthenia causing variants. And you see they are distributed all over the cell, and this model is rotating. So you see that these green dots are basically changing amino acids from the top of the protein to all the bottom and all the way in. The neonate alloimmune thrombocytopenia also has a distribution mostly and actually entirely on the ectodomain of the protein, because of course whatever is inside is not seen by the immune system of the mother, so they're only being outside what the immune system can see. Macroanisothrombocytopenia, they're, as I mentioned, mostly located on the inside and the cytoplasmic part of the protein. And the novel ones, which are the gray dots, are kind of distributed in all of the protein as well. And so then we went to do something that we scientists do, which is called sequence conservation analysis. So when you have a variant of your DNA, a way to address how deleterious or what the effect of this change will have in your DNA, we go back into something called sequence conservation. So if you can imagine, our DNA has been evolved and has been changing from when, and of course when there were insects, and all of us have proteins that have common pathways and functions. So when you look at amino acids in a species previous to human, and you say, okay, this amino acid has been maintained intact throughout the different species, right? So when we start in cows or rats and mouse, etc., we go in the evolutionary chain, this amino acid has been intact. This means that this amino acid is important because it has not been allowed to change. Any change has been kind of getting out of the genetic pool because somehow it might be very deleterious to the protein. So when you have amino acids that are very conserved, we think they are more important. But in amino acids in which nature has allowed changes, for example here, we think, okay, this has changes. Maybe this is an adaptation to the environment of specific animals, etc. But this amino acid might not be as important for the overall structure or function of the protein. So by doing this analysis, for each one of these novel variants, we analyze the degree of conservation of the substituted amino acid across the species based on a scale from 0 to 1, with 1 being the most conserved, so the more important for the structure and function of the protein. And we gave a score, we assigned a score to each one of these missense variants as an indication of the severity of the amino acid change. And we use a scale from 0 to 1, with 0 being the most severe. And this analysis takes into account the likelihood of an amino acid substitution during evolution. So here we see this analysis. So as you see, and here is the sequence conservation. So the higher you are, the most conserved that amino acid is. So it's likely that it's very important for the structure and the function of the protein. And here is just the residue number. So here you see that the Glanzmann variants, which are green dots, most of them are located up here with very conserved amino acids. And that's why when any of these amino acids are changing, they usually have a deleterious effect. So the change in the structure of the protein is prominent. The alloantigen ones, the orange ones, are more distributed like in amino acids that are not necessarily so important structurally or functionally. So they have a lower score, and you see some of them very, very low. And that makes sense with what is happening because this receptor is indeed expressed, right? The problem there is the difference between the mother and the child. And the novel ones are all over the place. We don't really know exactly. Some of them have low scores and some of them have high scores. So GT variants are mostly high and large circles showing the deleteriousness. So the results that we got as of here is that the majority of disease-causing Glanzmann Thrombasthenia missense variants involve highly conserved amino acids. So the scores were on top, near 1, and severe amino acid changes that are less tolerated evolutionarily. So 50% of the variants were responsible for GT had scores of less than 0.4. The alloantigens, when we analyzed this on the basis of amino acid conservation compared to Glanzmann Thrombasthenia, missense variants, the variants responsible for the alloantigens, were less likely to affect highly conserved amino acids or to exhibit severe amino acid changes. And 40% of the variants responsible for alloantigens have a mutation score of less than 0.4. And the novel variants, when compared to GT missense variants, was a greater representation of the variants, and they produce less severe amino acid changes. 30% of them had a mutation score of 0.4. So how will we use this information to predict of these novel variants? So from the GT we know, because we have information about the defect that they cause. But these novel variants that are in the population, we don't really know, and we don't really have a tool to assessing what defect they will cause. Will it be severe? Will it be Glanzmann? Will it be nothing? So there have been, because of the boom on next generation sequencing, there's also been a boom of tools to process this information and to try to understand this information. So there are different algorithms that have been created by different institutions in the US and around the world that have been developed to the purpose of analyzing genetic data and predict what these changes will have, the effect that they will have in the protein. So we use 3 of those. No, how am I going? Yeah. So we employed 3 different algorithms. And this study was done a couple of years ago. Now there are even more, and they are even better at predicting this effect on the protein. We use combined annotation dependent depletion, CAT, another one polyphen-2, and sorting intolerant from intolerant SIFT. So we anticipated the following. So confirmed and presumptive Glanzmann T mutants would be judged to have the highest likelihood of being deleterious. Alloantigens will have the lowest likelihood, and the novel variants will have the greatest range of likelihoods. So here are our results, and this is a little bit more technical. Here you see the results for CAD, for polyphen, and for SIFT. Because each algorithm uses different kind of mathematical models, the results are different. But in general, and you see in dark pink, the alloantigens, in green, the confirmed Glanzmanns, in blue, presumptive Glanzmanns, and in purple, the novel ones. So these results varied with the prediction tool, but in general, they were consistent with our expectations. As you see, Glanzmanns are higher on the rank scores, meaning they're more deleterious. The alloantigens are lower, meaning they are less deleterious, and the novel have a widespread around. So some of the novel variants have scores similar to those of patients with Glanzmann Thrombasthenia. So here we have. So we would predict that these specific variants are likely deleterious and likely will end up in producing Glanzmann Thrombasthenia. Some of the variants, and you see here in the green bracket, have scores much lower than those of the GT patients, and so they are likely not to be deleterious and not to be having any problems in the protein production and or function. But it's really not easy to decide where to put this cutoff, right? So we have a wide range of variation, and even with the most sophisticated analysis, it's still not easy to predict how these variants will be affecting the protein. So to conclude, we learned a lot about Glanzmann Thrombasthenia doing this analysis, and so the first thing that this you already know, Glanzmann Thrombasthenia is rare. So to find Glanzmann Thrombasthenia variants, one must sequence large numbers of individuals and such databases have been only become available recently. Establishing that the rare variants are not the result of sequencing errors using current methodology requires careful attention and new methods to minimize the risk of mistaken sequencing errors for variants of being, and these are being developed. Not even one of the reported Glanzmann Thrombasthenia missense mutations in literature was identified in all of the lists that we studied in this study. So they're all presumably have very low frequency, implying that they entered the population recently. Our study provides new insights into the frequency and potential impact of novel variants on alpha-2 beta-3. So in the 16,500 individuals studied, we found 114 novel missense variants affecting 10% of the amino acids in alpha-2 beta-3, and 68 novel variants affecting 9% of the amino acids in beta-3. Thus, we found that around 1.3% of the population studied harbors a variant on either alpha-2b or beta-3. The deleterious variants in alpha-2b beta-3 in the modern era are unlikely to be subjected to selective pressures, since the population continues to expand and humans usually mate with individuals outside their kinship. And as we enter the era in which many individuals will undergo next generation sequencing and have the possibility to know their genetic variants, it is evident that some individuals will want to know whether the variants they have are biologically or medically relevant or meaningful. We have a potential to provide a recent guess as to what is the likely impact of those variants. But even with the ability of these prediction algorithms and an advanced understanding of the protein structure and function, it is likely to be challenging to predict the functional consequences of a given genetic variant. And with this, I want to thank, so I'm presenting this, but I am representing a big group of scientists that work really, really hard to make this analysis. And this was a collaboration with Cambridge University and Rockefeller University. So this is the group of people that are behind all of this information and all of this data. And with this, I finish and I can take any of your questions. But my neck hurts.
Okay, Dr. Pichardo, I have two questions.
Yes.
And I'm not sure if you answered. I stepped out for like 2 seconds.
Yeah.
So the novel variants that you were describing, could those possibly present almost similar to GT? You were saying that they kind of were a little bit higher and showed with the...
Yes.
So it is possible for something.
Yes.
Okay. All right.
It is possible. So that's why this we try to really predict and see how wide is the variation, right? Because not all of them will result in Glanzmann. But some of them might. So that's yes. So some of them might result in Glanzmann Thrombasthenia.
And do you believe that, and I know you touched on it a little bit, but you believe that within the next, in the future, that being able to sequence and everything would help us kind of customize the treatment a little bit better to that patient and possibly have a better treatment plan?
I think in the future, and by efforts, for example, what Dr. Wilkos explained, right? So in genetic therapy, hopefully in the future will be more targeted. And now, for example, I don't know if you have heard of this CRISPR, right? CRISPR is going all over the news and all over the place. And this is supposed to allow you to edit specific regions of the genome, right? So hopefully in the future, with the advancement of this technology, combined with the knowledge, if you come, I really pray that this happens somewhere soon, that you come to a doctor and say, okay, this is my sequence. I want to fix this, specifically this change, and that will be possible with CRISPR, for example. Now we have the challenge of trying to fix, which is easier and better, because having this personalized genetic therapy, of course, is difficult at the part right now. But for example, the methodology that Dr. Wilkos is applying is to fix the whole gene. So this can be applied to all the different patients that have beta-3 mutations or alpha-2 beta-3 mutations. But technology is advancing really, really fast. CRISPR has completely changed and is now dominating the world of genetics. So hopefully that will, in the future, help us to have more personalized changes and applications for Glanzmann Thrombasthenia.
Thank you.
So I have a question. Can I ask a question?
Oh, yes, of course.
Yes. So there's a lot we don't know about Glanzmann, and we often wonder, are there other things that Glanzmann people with Glanzmann's have that you sequence the whole genome?
Yeah.
So you're able to look at all kinds of genes.
Yeah.
And sometimes there may be, is any form of Glanzmann people have, if they also inherited, let's say, the BRCA gene for cancer, you're able to look at that too, right? Or something for allergy, or maybe prothrombotic,
Yeah.
like the factor V Leiden mutation. So maybe you inherit something to make you bleed, but you also inherit something to make your blood clot at the same time. Would they offset each other, or there may be genes we don't even know?
So I think, so I did not do that analysis, but it is possible if you have kind of an analysis that is whole encompassing of the whole human genome. You can start saying how a specific variants in alpha 2b beta 3 can be maybe linked to a high frequency of variants in some coagulation factors or things. So I think it is possible. I did not do that. But it would be very interesting to actually, I think it's just so demanding right now computationally to do this kind of analysis. But it is possible that with the development of new methodologies, new algorithms, etc., we will be able to analyze the whole genome and see, and it is done for certain diseases, of course, how a specific change in one gene affects also changes on others. And this also makes sense because at the end, these genes are evolving together, right? So there is evolutionary pressure on if something is not working as should be, maybe we change something else to make that work as a whole. So it is likely that there are some associations and some changes with other genes. But as far as I know, nobody has done kind of that link study of different genes together.
Yeah, because I think there's so much unknown about Glanzmann, even people within the same family. Some have the same, you inherit different genes from your parents. Some people in the same family, one person bleeds a lot and the other person has GT and bleeds a little. Could be due to other inheritance. I don't know, but we often wonder, and I think that this technology could help us to learn more about GT and why we see differences and maybe certain things. So it's a very great, powerful technology.
It is. I think genetic sequencing is incredibly powerful, and it will for sure transform the way we think about disease and treatments, hopefully. Yeah, for sure.
All right. If there aren't any more questions. First off, guys, a round of applause for Dr. Wilcox and Dr. Dusado.